Welcome, everyone. My name is Lynn Mullen, and I'm a senior scientist with QIAGEN's Bioinformatics. Today's focus is on how to format and upload your data set into IPA so that you can perform the different analyses and harness the number of capabilities that IPA has to help power your research. This webinar is suitable for new users of IPA, although more experienced users might pick up some information that could be helpful for them as well. And this is scheduled to last for 45 minutes. I'll spend about 35 minutes or so presenting and then open it up for questions for the remaining amount of time that we have. And this is just a note that the software presented in today's webinar is for research purposes only. And this webinar is the second in this webinar series. So last week, we held a webinar during which we demonstrated the results you would receive from an IPA core analysis. And then next week, we're going to end the series by talking about how you can further explore your results using some of IPA search and pathway tools. And these features can be used to test different hypotheses, both with and importantly, even without data. And I'm going to tell you more about the date and the time of this upcoming webinar at the end of this presentation. Okay, so first I'd like to provide you with a brief overview about the webinar. So I'm going to show you how you can format your data, but first going into the example analysis data set that we're going to use today. And then I'll show you more specifics of how you can format your data, a bunch of different types of data that you can use. And then I'm going to show you, going into IPA Live, how you can upload your data and then how you can start an analysis like a core analysis. And then finally, I'll answer your questions. Okay, so let's start by going ahead and looking at the background of the example data set we're going to be examining today. So you might recall this slide from last week's presentation. So we're going to use that same data set so you can see first how you format and upload it, and then last week's presentation, you can then look at the type of results that we can see here. So if you recall, gemfibrozil, it's a drug that's often prescribed for patients who have pancreatitis, who have high levels of triglycerides in their blood, high cholesterol. And it's an agonist of PPAR alpha, and that's highly expressed in the liver and brown adipose tissue. So in this study, we took it and downloaded it from Geo. It was an RNA-seq study from 3 rats that were treated with gemfibrozil for 7 days, and then they were compared to rats who were orally gavaged with corn oil. And what we did is we processed this data, calculated the levels of differential expression between the treated rats and the control rats. And from those calculations, we got fold change values, p-values, false discovery rates, and RPKM values. Okay, now with that, let's go ahead and look at the data set. So I just want to go over the most basic format. So first of all, you can use an Excel file. You can also use a text tab delimited file. And if you're using an Excel file, you want to make certain that all of your data is on the first worksheet that you're going to upload into IPA. You can have other tabs, it's just that IPA is only going to be looking at the first worksheet. The other thing you want to keep in mind when you're using an Excel file is that you don't want to use the .csv extension. You want to keep it as .xls or .xlsx. And IPA requires that you have one column be assigned as the identifiers column. And what I mean by an identifier, it's a unique annotation of a molecule that you can find from either vendor or public databases. So for example, Mirabase or PubChem, EntreGene, RefSeq, and UniProt, and many more. Now, if you can have just one column and put in different types of identifiers, and IPA will look at that while it's scanning the data set, and it will be able to make smart assumptions about the type of a number of different identifiers you have. But you can also place each ID type into a separate column if you wish, and you can have up to 5 separate columns. But it's really important that your leftmost, this first column, does contain the identifier. So you can have one column with identifiers here, but you can also have identifiers, say, in column J or K if you wish. but you do need to have that first column contain an identifier. And I'm going to tell you how you can tell IPA which columns contain your identifiers when we go live into the program. Okay, so another thing you want to keep in mind is you can only have one header row, and you can include as much information as you like here, so be as descriptive as you like with these different headers. And IPA isn't going to calculate the full changes, the p-values, RPKMs, et cetera. So that has to be done before you upload that data into IPA. And that can be done in programs like GOR. It could be done in our CLC workbenches, among many different other software. And if you have replicate samples in your data set and you want to take the averages, you want to treat them as a group. So you have to either take the averages or the medians, and you're going to place that into a separate column within the spreadsheet. And you'll tell IPA when you upload it, this is the column of the averages or medians that I want to include. So you can include all those replicate sample information, but you can't map each replicate unless you want to treat each replicate as its own observation. And what I mean by the term observation is that it's a comparison of different groups within your experiment. So for example, an observation could be a treatment versus control, like I have here, or it could be a disease versus a normal tissue. And now this data set only has the one observation. So it's the gemfibrozil versus the control rats. But I want to go back to my slides to give you another example of a more complex data set. So what we have here is we have a data set that has 3 different observations. So this experiment came from a study that examined the development of cardiomyocytes over time, starting with the embryonic stem cells. So here we have the mesoderm, MES, versus the embryonic stem cells. That's one observation. The second one is CP, or cardiac precursor cells, compared to those embryonic stem cells. And then the third observation is the developed cardiomyocytes compared to those embryonic stem cells. So you can have up to 20 observations in one data set. And importantly, you can see here that I have 3 different types of data measurement values. So I have full change, RPKM intensity, and FDR. And I have that for each one of my observations. And you can have up to 8 different data measurement types for each observation, all in the same data set file. And I want to tell you now a little bit more about the type of measurement values you can upload into IPA. So here's a list of data values you could use if you're doing expression or phosphorylation data. So our expression values are here, and the expected values are here, and as well as phosphorylation with their expected values. And you can see that a lot of them are exactly the same. So we have ratio here, ratio here. There's 2 different differences though. You can have a phosphosite option, and that allows you to include the actual phosphorylated peptide sequence. You can include a residue number or any combination of different texts and numbers to describe that site. And then you also have the option to use data that doesn't fall into any of these categories, and you can just classify that as EXPR other, as long as you have it normalized around 0. And then there's also an option to analyze genetic variant data, so variants that are classified according to their loss or gain of function, and also those that are classified with the American College of Medical Genetics and Genomics classification, whether they are benign or pathogenic. And you can see here that we use categorical values for these interpretations. And the note option isn't a data type per se, but it does allow you to keep track of a molecule with your own notes. You can have up to 256 different letters or numbers for each one of those. And what will happen is when you look at your analysis and you hover your mouse over that particular molecule, you'll see the note appear in the analysis. And one common question that we get is, what if I don't have any data measurement values? So I have a list of, say, genes or proteins, chemicals that I think are potentially interesting that I got from some different experiment or from literature mining, for example? And the answer is you absolutely can just upload those molecules without any values whatsoever. You just won't be able to get information like the z-scores to measure predictions of activation or inhibition. Okay, so let's go back now to finish up the formatting portion of the webinar. I just want to make a couple more comments. So first of all, you shouldn't have any blank rows in your data set. And you have to be really careful about special characters like pivot tables and the like, because that can cause problems with your data upload in IPA. And you'll also want to carefully look over your data, because sometimes if you convert, say, a text file to an Excel file and you have a gene like Sept7, for example, IPA is going to think that's September 7th and it's going to make that conversion. Or you'll see a calculation for minus infinity and it will turn into a formula error. So instead, what you can do here is you can put an apostrophe, and let's say Sept7 will turn into Sept7. Okay. Now there's some exceptions to the rules that I've outlined. So if you're going to do, say, a batch upload, you want to upload a whole bunch of different data sets at once, then you're going to need to include metadata, and that'll be on at the very beginning of the spreadsheet, and then underneath that you'll have your actual data measurement values. And we have more information about that in our online help manual, so if you're interested in doing that, you can look up the exact structure there. And there's one other exception to that, and that's if you're using cufflink software. We do have the ability to allow you to directly import that into IPA. So let me just talk about that for a little bit. So you can upload those files directly from cufflinks. And please note that you are going to have to take the files directly from the output to the file. So you can't make changes to it. And when you set up that, when you start to import, you're going to see this pop-up window, and it's going to ask you to eliminate the different genes that have unacceptably low FPKM values in both the experiment and the control. And by default, you'll see that it says 10, but you can change that to whichever number that you like. And then in cases where the cufflink specific IDs are used instead of gene or transcript specific IDs, then IPA is going to ask you for a corresponding FPKM tracking file, because it needs to make the conversion from the cufflinks IDs into identifiers that it recognizes. Okay, so now that I've shown you how to format the data, what I want to do now is go into IPA and show you how you can upload it and then how you can start your analysis. So let's go into IPA. You might recognize this welcome screen from last week's webinar. And last week, I just showed you features for how to start the analysis. Today, I'm just going to focus on the features for uploading your data, and I'll go over some of the other things on this welcome screen next week when I talk about the different tools that you can use with or without data. So first of all, starting up here on the left, we have a file. You can go and upload your data set here, or batch upload, like I mentioned earlier. A really faster way of doing that is just clicking on the analysis, and that will actually give you a button where you can upload that, and I'll show you how to do that in just a minute. And you can also go to new, and that will show you many of the different options there. And then you can also click on these analysis wizards. So click on Analyze Dataset, and that will give you the option to either pick a data set that you've already uploaded or to upload from scratch. And your data sets, once you upload them, are going to be in the My Project section. So here we have the data set files, and that's where your data set will go. Okay, so I'm going to start from scratch because I already have data sets, lots of data sets, but I want to start from scratch so you can view how to do this. So here you can see this window. It shows me everything in my projects folder, but I also have this upload button. So we're going to go ahead there, and it will ask you to navigate to the file. So I called it very simply Gem versus Control, and click on Open. And then you can see over here we have these workflow instructions. So this includes a lot of different screenshots and information in case you've forgotten how to do it. I'm going to go ahead and close that out right now. Okay, so I'm going to start from the top and work my way down. The first option is called select file format, and you can see by default we have flexible format. There's two other options, but we don't recommend using them unless you really want to make a comparison between something you did like 10 years ago and now, but just sticking with flexible format works for just about everybody. Contains column header, so IPA will scan your data set and it will tell you if there's a column header or not. And the select identifier type, it just asks you to assign your ID columns. It will automatically do that for the leftmost column, but if you have additional identifiers, it won't do that at all. So you have to pick which identifier type you have in these additional columns. And then the array platform used for the experiments. So here you can choose the platform from which your data was generated if you had a microarray data. And I don't have microarray of RNA-seq, so I can safely choose not specified applicable. If you have proteomics or metabolomics, then you can also choose this option. Okay, so now let's go down here and take a look at our data in the raw data view. You probably recognize what we see here. It's all grayed out, but once I start to make selections, it'll become active. The first thing I want to point out is this Infer Observations button. So you can safely use this if you have a data set just like mine, where you just have a column of identifiers and just the data values. But if you have, say, a bunch of replicates, or you have your identifier column here and you have an identifier column over here, then you're not going to be able to use this Infer Observations button. Now, I could use this today, but I don't. I want to show you how to do it from scratch. So instead, what I'm going to do is I'm going to go ahead and click on ID. And so IPA has already successfully determined that I have ensemble identifiers. And you can see here, this is all the different identifier types that you can use. So RefSeq, PubChem, KEGG, Hugo, Entree gene, and much more. And I mentioned earlier, you can have more than one identifier type in the same column. So this is a really good way to go ahead and check and make certain that, yes, IPA has called that correctly. Okay. You can see that after I made that selection, that this data set summary went from 0 to about 21,000 different genes. So the numbers are different between the raw data and this. So if you click on this, you can see a summary of what's going on. So we have about 21,000 mapped. So in other words, IPA recognized that these were identifiers and that they were mapping to genes that are in the knowledge base. And then we have unmapped. So these are a bunch that weren't recognized by IPA. And there's a variety of different reasons for why an identifier can be unmapped. So for example, a gene or protein ID doesn't correspond to a known gene or protein product. One of the most common reasons is because the unmapped identifier is from an older version of a database. So in these cases, you're going to have to access the newest version of the database to find that updated information. And you can see that information by going to show details, and it will show you the mapped information and then here the unmapped. So if you see some unmapped, you might want to go ahead and look at the various databases and see if you can include more and re-upload your data set. All right, let's go back to the raw data view. And now what I'm going to do is I'm going to tell IPA which columns contain the data that I want it to analyze. So I'm first going to choose this column and I'm going to select observation 1. And you can see now we have a new drop-down window that appears, EXPR fold change. IPA has looked at the column header. If you have something like FC fold change, it will map it as fold change. If you don't have a column header, it will still scan it and it'll try to make its best guess as to what information that you have here. And it's important to note, you can see when I do this drop down window that these are, you probably recognize this from the different slides. One thing that happens if you have phosphorylation data is it will still automatically choose EXPR, and it's important that you instead go down and choose the phospho option. That makes a big difference with the calculations of the z-scores. And there's two other options that I didn't go over in the slide, and one is called absent and override. So absent tells IPA that you want it to ignore a certain molecule in the data set. So you already know a particular molecule isn't important to you, so you don't want it included in the study. And then override is the opposite. It tells IPA to include the molecule, even if it didn't pass any cutoffs or filters that you're later going to apply when you set up the core analysis. Okay, so now what I'm going to do is I'm going to choose observation. Note that observation, again, does not mean a data set measurement. It means the comparison between your experimental group and your control, right? So all of these are going to be observations. We just have different data measurements for that same observation. You also notice it says adjusted p-value here. So again, it said, oh, p-value. What I like to do just for consistency, just kind of remember later on what I did when I'm looking at the analysis is change that to the false discovery rate. And one other thing that I recommend doing is editing the observation names. I like to do this because often you're going to want to share your data with other people, or maybe you want to go back and look at your analyses, a month later, for example. And also when you're looking at your analysis results, it's just so much easier to just look at them and not have to remember what observation 1 and 2, et cetera, stands for. And that's also particularly helpful when you're comparing these observations side by side, like in a comparison analysis. So to do this, you just click on the edit observation names. There's a little drop-down menu you can use, but I'm just going to type gemfibrozil versus control, and then click OK. And then that automatically changed the observation names. And then when you're done with that, you can click Save. And we have this new window that pops up. I think we included this like a year ago, and it kind of freaked people out who had been using IPA because they thought, oh, what did I do wrong? I've been doing everything exactly the way I did before. Well, what we did was we included a new metadata tab. So it's just saying, hey, you don't have metadata, you might want to use it. If you don't want to use it, you can just click OK and move on. But I want to show you that metadata, so I just clicked cancel. So here's the metadata tab. The reason that we think it's really helpful and we included it is, first of all, when you go ahead and do searches later on in that project manager, it's really nice to have the metadata option, and you can go ahead and it will, if you have analysis match license and you're looking for everything associated with coronary disease, for example, then it will find that, and it can also find your data set that was associated with coronary disease. And the other option is that it's good for is these are the same key terms that we use in analysis match, so it's a lot easier to filter your data set if you stay consistent. You can see that there's a lot of different terms here, so we can do something like, okay, I'm interested in case treatment. So I'm going to double click on this value, and then I can just type in the treatment there. Okay. You can also add your own key terms using this Add Custom Field button. And then we have a hide rows with empty values. So if I only want to see, well, what have I entered? There's a lot of information here. I just want to see what I've entered right now. You can click on the hide rows with empty values, and boom, there you go. All right, so after that, now we can go ahead and hit save. And then we can go ahead and either create a new folder, or I'm going to navigate to, where is it? It's far down here, IPA webinars. You can type in notes. You can change the name of the file. I'll keep it the way it is and click on save. Okay, so it already exists. So I'm going to go ahead and just add a 2 to that. And now we're going to go ahead and upload the dataset file. It will be stored in that dataset folder associated with your project in the project manager. So this will, because I chose core analysis, I didn't go to upload dataset. I just chose the core analysis option when I first began. It's automatically going to take me to the steps that I need to take to go and perform the core analysis. So I don't have to go back in, double click on the dataset, and then start the core analysis. So what we're going to see is we're going to see soon a window that will pop up, and it's going to show me, first it's going to ask me what type of analysis I want to perform. So do I want to perform an expression analysis? Do I want to do a tox analysis? So if I've chosen EXPR, my first option is expression analysis, but if you recall from last week, you can do with expression data a tox or metabolomics analysis. If you have a phospho fold change, you've assigned phospho values, then you'd only get the option for the phospho analysis, and same with variant information, it would only say variant effects. And then this measurement type here, you need to select, you need to select either the expression or the phosphorylation data measurement value that you used, because you can only use differential expression or phosphorylation data when calculating your z-scores. And again, those z-scores tell you about whether a pathway or disease, et cetera, are activated or inhibited. And it also tells something about the coloration that we're going to use. So I have all these different options that I can choose. If you accidentally choose a statistical value instead of an expression, then it won't calculate any z-scores. So that's something that I've seen before, where for whatever reason, p-value popped up there, and then someone's like, why am I not getting z-scores? And that's the reason why. Okay, so here's the create expression analysis. So you can see here we have a bunch of different options, and then we have some cutoffs, and then we have a preview data set. So I'm going to work my way from the top, and we're going to start with general settings. So the first option here is the reference set. So this allows you to choose the background set of molecules against you're testing your molecules against that background to determine the statistical significance of particular pathways or functions, etc. So if you don't know where your data came from, you can just safely choose the Ingenuity knowledge base. We have options if you're doing metabolomics that you could just do endogenous chemicals, or you can do a combination of the two. We also, if you had chosen your array on the previous section in the data upload, you would see that listed here, but if you forgot, you can choose the array here. And then finally, we have a user data set option. So this is an option that you should use if you haven't tested, say, the whole genome, or you're using, say, a targeted RNA panel. So let's say that you tested 500 genes that are involved with immunity and inflammation, like one of our targeted RNA-seq panels. If you chose the Ingenuity knowledge base, the genes only option, then you're biasing your results, because you have only 500 genes that you've tested, and they're all associated with inflammation and immunity. So imagine if you ran your core analysis with just the genes only and you tested them against the whole genome. Wow, inflammation and immunity are really highly significant. And that's not the case. So instead, what you'll do is you'll select user data set. Importantly, you do need to set cutoffs to determine which of those molecules are significantly perturbed, and then you can run the analysis. But I'm going to keep it at genes only. And by default, the relationships for the networks and upstream regulators are going to be direct and indirect. So direct interactions require that two molecules make direct physical contact. An indirect relationship example would be a growth factor that causes a change in the expression of another protein. Now, in the network section, you can see that by default, we include endogenous chemicals, and that's just to maximize the number of relationships. So even if you don't have endogenous chemicals within your data set, you can include this option. And we have these values for the molecules per network, but you can change that to make more. The networks per analyses, you can change that as well. We chose these numbers by default because we found that it gives you the maximum numbers that can be used to view easily on a screen, but it gives you as much scientifically relevant information as possible. And then by default, this causal networks is unselected or deselected. Even if you have advanced analytics, you'll see this active. If you don't, you'll see it grayed out. I highly recommend checking this option because first, causal networks, they're looking for master upstream regulators. It's really a great feature of IPA. But also if you have the analysis match license, as I mentioned last week, you'll get blank columns for the causal networks, and that's really going to affect the matches. You're not going to get as great matches. And then if you want to, you can score the master regulators against your data set, but you could also say, hey, I'd like you to score some master regulators against a particular disease of mine or a particular chemical or gene. You can enter those here. It's just like the search engine here, which I'll talk about next week, and you can add up to 50 of those right here. The node type, you could go ahead and you can change different molecules. So if you want to eliminate certain types from being analyzed, you can do that. We have a bunch of different other options. So the confidence refers to microRNAs and more. The advanced button, so this will allow you to change how the duplicate identifiers are scored and how you can color the different nodes. And then let's say that you've made a lot of changes here and you don't want to keep doing it over and over again for your upcoming analyses. You could just save what you have here selected as a default. I'm going to go over the set cutoffs in a little bit, but I just want to draw your attention to this table down here. It's our data set, but IPA has also included information here about the name, the symbol, and what it knows about those particular molecules, like its location, whether a drug targets it, and more. And you can see here that we have the metadata. All IDs is what our raw data view looked like. The unmapped and mapped, again, I went over that before, but you might notice we have analysis ready tab. That's new. And analysis ready, the number is different. We've got 5,000 fewer genes than in mapped. So the reason for that is because we have a lot of duplicate identifiers for the same gene. So IPA is only going to accept one gene, protein, etc., for one identifier for each one. So you only get one identifier, and you can see which ones had duplicates because it'll have a flag that says D right here. You can see that legend for the other options as well. And you can see when I select things, I can create a new data set, add to my pathway. I can navigate over here to a particular molecule that I'm interested in, and more. Now you can also see here the number of analysis-ready molecules in this observation. This is a lot because what we do is we do recommend that you set some sort of cutoff for your data, especially if you're using the user data set as your reference set. So this is because you're going to want to analyze the molecules that are most highly perturbed in your experiment as opposed to every single gene or protein or chemical that's available. And if you recall, I have these 4 different measurement types, and here's the range. And here's where I can set the different cutoff values. So we don't have a cutoff that we recommend. We don't say you absolutely must put in 1.5 for the cutoff. These are the values that I like to start with. You can actually run as many analyses with different cutoffs as you like, and then go ahead and compare the results. And you can also, of course, here, if I want to choose a 3, I can do that. So we're going to keep it there, though, and hit recalculate. So this gives me about 1,200 analysis-ready molecules. I like this number because what we recommend that you do is that you keep it somewhere between 100 to 3,000. That tends to give people the best results. Okay. And so now what I'm going to do is run the analysis, and it's going to actually say, hey, here's a timestamp. I like to add in what I just did for my cutoffs because later on when I'm trying to see, okay, I ran different analyses. What did I do? What was this cutoff? What was this one? And I have the timestamp. So I like to add all this information here. And then I can go back later. So the reason I like to do that too is, let's say that I've run 3 different analyses with different cutoffs. I'd have to open up each analysis and look in the analysis settings to see what happened. But here, now I know automatically what I did. So when you click on that OK, you get that window, and you can always close it. And then here I have this little clock icon, and this is now in my analyses folder. So this means that it's in the queue and it's waiting to be analyzed. And when it's actually going to get started, you'll see this little running circle that appears. And then when it's done, you'll see that it turns into bold. If you're logged into IPA, you'll also see this little orangey yellow bar that'll pop up at the bottom. And you'll also get an email that will be sent to the email address that you used to log in to IPA. Okay, so with that, I'd like to end the presentation by giving you a summary of the best practices for formatting and uploading your data into IPA. So first of all, you should make certain that you have one ID column that's in the leftmost column, that you have no empty rows in your Excel or text tab delimited file, that you have up to 20 observations. So in other words, those different time points, different phenotypes, comparisons, and so on. And you can have up to 8 different data measurement types, but again, you could have 0. You could just upload a list of molecules without any measurements. And then any calculations for data measurements have to be made outside of IPA. And if you're importing your CuffDiff files, you have to take the file directly from the output of the CuffLink software. You'll want to label your observations so you can go back to the analysis later and easily make sense of your results. And I suggest adding metadata, which could be useful for later when you're searching for different analyses or data sets, or it could be used for to help you with matching up and filtering in the analysis match feature. And you also want to use your different filters and settings in such a way that you're stringent enough to gain meaningful information, but give yourself enough flexibility that you don't lose important information. So again, we recommend that per observation, you have about 100 to 3,000 molecules. If it's much larger than those values, then you're essentially asking IPA, well, what does the genome do? What does the proteome do? Instead of focusing on your specific question in your study. So I hope that you have a better understanding of how to format your data and upload it into IPA. And I'd like to invite you to participate in next week's webinar. So it's going to be held a week from today at the same time. So here I'm going to show you how you can use the different interactive tools in IPA to further test hypotheses. So you're going to be able to use these tools to expand upon the results that you're getting from your different analyses, or you can use them by starting from scratch, just with a question. No data at all. And if you'd ever like to contact us, here are our phone numbers. You can email us at TS, which stands for technical support, bioinformatics@QIAGEN.com. We also, if you're interested in getting an upgrade to your license, you want a license or you want a free trial, you can contact bioinformatics sales@QIAGEN.com. And then here are our different websites. And also, I mentioned earlier that we're recording this webinar and it will be made available to you later, but you can also in the meantime watch some tutorials at tv.QIAGEN Bioinformatics, and there's also some specialized webinars that have been recorded in the past. Okay, so I hope this has been helpful. I hope again to see you next week at this upcoming session. There's a lot of cool tools in IPA that I hope you can, that I can share that information with you. Okay, well, thank you very much, everybody. I really appreciate the time that you've taken today, and I hope you have a great rest of your week. And for our Americans, happy Columbus Day. Okay.
QIAGEN.
Sample to Insight.